Our lesson today is on proteins. Proteins are molecules composed of chains of amino acids. So your textbook, I think, actually mentions there's more than this, but there's estimated to be 100,000 or more different types of proteins in humans. Each has kind of a special function. Your cells make them to complete a very specific task or to serve some very specific function. One example of a protein is called an enzyme. These are proteins that promote specific chemical reactions. So they make reactions happen faster, or ones that never happened at all can actually happen because of enzymes. So it's ways for your cells to create and perform reactions that otherwise would not be happening. Some proteins are for structural supports, things like keratin, right, things that's on your hair, horns in animals or nails. Some allow for motion, so actin and myosin. These are found in muscle cells. They allow the muscles to move back and forth. Some assist in transport, so like hemoglobin, which is in your blood, helps carry around the iron that's in it there. So there's lots of different functions for these things. You might remember earlier we talked about how things that end in IN and things that end in ASE, right? These are typically the giveaway in the spelling that this particular thing is a protein, right? That's what you're looking for. It ends in IN or ends in ASE. So proteins are polymers of amino acids joined by peptide bonds. Peptide bonds are the special name for the bonds between amino acids. They're still linked together by dehydration synthesis, just like all of the other things we've talked about that are linking together, dehydration synthesis. All amino acids have the same fundamental structure. The backbone of this is going to go nitrogen, carbon, carbon. Okay, and it's just going to repeat in that order, nitrogen, carbon, carbon. You have a central carbon atom bonded to a hydrogen. This middle one here is going to be bonded to a hydrogen. A nitrogen-containing group, so this is going to be NH2. One of those hydrogens is going to disappear when it forms a peptide bond. A carboxylic acid group on the right one, so that's going to be double bond O, single bond OH. And then in the middle here, we have this R group. Okay, and this is a group that changes. So everything else about the amino acids, the nitrogen, carbon, carbon backbone, that's not going to change. This R group, right here, okay, that I've drawn, and right here how they've drawn it, that is variable. So it can be different things, and the cell will make that to do specific functions. So if it wants it to be hydrophobic or hydrophilic or acidic or basic, whatever it needs it to be, it can add that particular R group there. So that's what that function is. But the backbone of it is still just this nitrogen, carbon, carbon, over and over and over again. We'll see how these form bonds with this hydroxyl part of the carboxyl group and this amino group here later on. So there are 20 amino acids commonly found in the proteins of organisms. And it's pretty cool because we'll find out later on that that is universal, the way that these things are coded, which is pretty cool. The R group gives each amino acid distinctive properties. That's what we were just saying on the previous slide. Some are hydrophilic and water soluble because their R groups are polar. So that one part of the chain there is going to be polar. Others are hydrophobic, which means they're going to be non-polar, right? Non-polar. Okay, it's right there above it too. Non-polar R groups make it hydrophobic. And then we have something called cysteine, which we've actually talked about before, which contains sulfur and can form disulfide bonds with the sulfur of another cysteine molecule. This is going to lead to some secondary and tertiary structures, which we'll see here in just a few minutes. But cysteine is going to be pretty important for these amino acids because of these disulfide bonds. Okay, so here's an example. You don't need to write these into your notes, but here are a few examples of amino acids. Notice that this part of it is the same for all of them. The only thing that's different is this R group. So here's one called glutamic acid, aspartic acid, phenylalanine. We'll talk about that one later on as well. Leucine, cysteine, which is the important one that can form that disulfide bond there with that sulfur. So a lot of different variety here in terms of what it can do, right? This one's got a carboxyl group that can make it acidic. This one's got a carboxyl group that can make it acidic. This one looks to be pretty nonpolar. This one looks to be pretty nonpolar. This one's got that sulfur for disulfide bonds. So can have very specific functions just because of those R groups. Amino acids are linked together by dehydration synthesis. We've talked about that before. Just like polysaccharides and lipids, a hydrogen from the NH2 group breaks off with a hydroxyl part of the carboxyl group. Okay, and we'll see what that looks like here on the next slide. This bond is called a peptide bond, and the resulting chain of 2 amino acids is called a peptide. And if we get a whole bunch of them together, it is called a polypeptide, right? So proteins are called polypeptides if you get 50 or more peptides. A protein consists of one or more polypeptide chains. Okay, so that's what these are. So we've been talking about how you build up these molecules. Right? We've talked about carbohydrates coming from monosaccharides. We've talked about lipids coming from glycerol and fatty acid chains. Here for proteins, right, it's a linking together of amino acids to form peptides. Peptides all linked together form a polypeptide. So here's a picture of what happens in this reaction. So remember, it forms a long chain, and I told you that the backbone is a repeating nitrogen, carbon, carbon backbone, right? Nitrogen, carbon, carbon, nitrogen, carbon, carbon. So when this whole thing links together, that's what the backbone of this is going to look like. It's dehydration synthesis, which means that, right, we produce water, we produce H2O when this is going on. So how does this work? Well, we're going to link them together in a chain. Notice, if here's our carboxyl group, there's the hydroxyl OH part of this, and the amino group over here. Well, this hydroxyl part of the carboxyl group and this hydrogen from this amino group are going to split off and give you water, right? So that's what's showing you right here. This, okay, let's change the color here. All right, gives you the water. That's where the water is coming from. And the result then is that this carbon right here actually links up to this nitrogen. Okay, so that carbon goes onto that nitrogen, and we get this repeating nitrogen, carbon, carbon, nitrogen, carbon, carbon chain. Okay, so we can just keep doing more dehydration synthesis, right? You just line up another amino acid right here, okay, with the amino group on this side, okay, and then you can just get more water off that dehydration synthesis, make the chain longer, keep that nitrogen, carbon, carbon backbone going. So as far as structure goes, proteins can have as many as 4 levels of structure. These are primary, secondary, tertiary, and quaternary. The primary level is just the order of amino acids. Okay, so like the glutamic acid or phenylalanine, all right, whatever the particular order is, that is the primary level. The sequence is specified in the organism's genetic code, right? We've talked about where do we get all this stuff from? Well, you get it from your DNA. The DNA actually codes for proteins, and if you have mistakes in your DNA, you have mistakes in your proteins, and your cells don't function optimally. The sequence of amino acids causes polypeptides to assume a simple, repeating secondary structure. So depending on what their primary structure is, the order of amino acids, you can get some three-dimensional structures, okay, secondary structures. So things like a helix or a pleated sheet, okay, and if you don't have those in your notes or an example of what they look like, you should draw those in there. Helix and a pleated sheet, which we'll see here on the next slide. Okay, so for helix, proteins like keratin form a helix. Okay, so you're going to see that it actually forms, right, kind of this structure that keeps going around and around and around. The pleated sheet, right, so an example of this might be something like silk, contains polypeptide chains that repeatedly fold back upon themselves with hydrogen bonds holding adjacent segments of the polypeptide together to form a pleated sheet. Okay, if you know what pleated pants are, it means you got to fold, okay, on the front of them. So this kind of folds over, and we'll see what this looks like. Okay, so right here, this is an example of a helix structure, right? So you can see how it kind of wraps around itself. It's going to be held in this helix shape, okay, by these polar bonds here and here. Okay, we have slight attraction between these, which is going to hold the molecule together in that helix structure. For a pleated sheet, okay, so you can't necessarily tell by just following the atoms here, but it kind of bends back into the page and then back out of the page, then back into the page, then back out of the page, all right? And we have these repeating structures here, right? This is what our polypeptide is. Keep going this way. But notice we are linked this way now rather than in a helix pattern. We're linking polypeptides across from each other, okay? They kind of go back and forth. And we end up with this back and forth pleated sheet pattern, okay? So that's what silk If you look at silk, that's what it looks like on an atomic level. So, in addition to their secondary structures, many proteins are contorted into tertiary structures. So this is how proteins shift their three-dimensional shape depending on their environment. Okay, so even you've got the primary level of the order of amino acids, you've got the secondary level of what their 3D structure looks like. Tertiary means we can change shape depending on what type of environment we're in. So if a protein, say, has hydrophilic and hydrophobic parts to it, it will be able to change conformation based on whether it's in water or not in water, or whether it wants to dissolve or not dissolve. Okay, and then lastly, quaternary structures described in two or more polypeptides linked together by hydrogen bonds, disulfide bonds, or positive-negative attraction. So an example here is hemoglobin. Okay, notice it's not made out of just one long chain. It's actually multiple chains, okay, that all seem to have the perfect shape to fit together. And in the middle of that, as you can see, are the iron plus 2 ions, the Fe plus 2 ions. This carries the iron through your blood, this protein here. So that's an example of a transport protein. The shape of a protein and its order of amino acids determine what processes it does or which function it serves. So the textbook mentions that proteins are the most diverse group of biological molecules. They can have so many different functions. And it's because we can order, there are 20 of them. It's kind of like an alphabet, right? We have 20 amino acids that we can put in whatever order we want, and we can make these, 50, 100, 10,000 if we wanted to, amino acids long, and you can end up with different functions, okay? It's almost like creating a language. If a protein loses their shape, they become useless. They don't do what they're supposed to do. They can't perform the task they were designed to do. So if you change like an amino acid out, swap it for something else, you might ruin it. If you change the environment that it's supposed to be in, you might make it non-functioning. So you got to keep them in their right shapes as well, in addition to the right order. So what happens when you mess that up? This is called denaturing a protein. It means you're destroying its three-dimensional shape and thereby its functionality. It no longer works like it used to. You do this every time you cook anything with protein in it. You are denaturing it. You're basically heating it up to break down those proteins, which makes it easier to digest. Okay, that's why it's easier to digest fully cooked meat than, say, raw meat, because the proteins are denatured. Same thing happens when you're cooking an egg. Okay, what you're doing is you're denaturing the protein in the egg, which makes it cook. Anything like that, anything you're heating up basically that has protein in it, you are breaking down that 3D shape and making it easier to process, easier to digest. Humans consume the proteins as denatured after being cooked. Right? What happens if proteins become unfolded naturally in our body? So there are some diseases called prion diseases. Prion diseases, or prions. They are really horrible. It's an example of a disease that messes up proteins in your body. These are viruses. A lot of these are viruses. You have prion viruses, and then some of them just naturally occur in human genetics over time, which are also just really sad. An example of this would be something like mad cow disease, which you may have heard about in the past. It hasn't been around, any time real recently, but there was a real concern a number of years ago about mad cow disease, because what would happen is people would eat these pieces of meat and then they would start seeing symptoms of dementia and Alzheimer's in very young people. What ended up happening was they tested that meat and found out the meat was full of prions, which are basically proteins that are currently denatured, and they're denatured in a way that actually denatures other proteins around them. So it kind of amplifies denaturing. If you get one denatured protein that's a prion, the prion will then affect other functioning proteins around it and shut them down. So they stop functioning. So that's what mad cow disease would do. It was a virus that initially caused that there, and if you were ingesting it, then you'd end up with these prions. Then this is really what leads to things like dementia. If you have a grandparent or loved one who's elderly, may have dementia, or, if I can spell it right, Alzheimer's. I don't know if I'm spelling that right there. Okay. What ends up happening is that they have prions forming in their brain. The proteins become prions, and they stop the other proteins in the brain from functioning normally. And over time, that leads to weakened brain function. poor memory loss, things like that. It's really kind of a horrible way to go. But these are prion diseases is what those are. So like if you're trying to, create a cure for Alzheimer's and dementia, or even mad cow disease, like what you would need to do is find a way to stop the prions from affecting the other proteins around them, from denaturing the other ones, the good functioning ones, which is not the easiest thing to do. Anyways, that'll wrap up our lesson on proteins.